Good day, class. In this video, we would learn about domain Archaea. So to start our discussion, let's look at this meme from the Amoeba Sisters. So the daughter was saying, "But father, I love him." And the father was like, "He's in a completely different domain." Now how come that the course of true love did not really run smooth between Archaea and bacteria? As we have learned before, after Carl Woos has discovered the new domain, which is Archaea, the former kingdom of Monera was split into two, Archaea and bacteria. So in this video lecture, you're expected to first understand the basic structure of cells belonging to Archaea. Second, relate the various structures to the different adaptations of Archaea. And third, realize the ecological and economic importance of this group. So to start our lecture, let us first look back into this phylogenetic tree from Campbell. Here you will now see domain Eukarya, domain Archaea, and domain Bacteria. Notice that domain Archaea is now divided into the Euryarchaeotes and the tax superphylum. However, in a recent study by Baker and others in 2020, aside from the presence of Euryarchaeotes, the tax superphylum is also present, and other superphylums were proposed, including Asgard and the Deepan superphylum. We will be talking more about these different groups as we go along the video. Now, before diving deeper into the phylogeny of Archaea, let us first look at the characteristics of Archaea. First, they do not have any peptidoglycan layer, and they could now either have a lipid monolayer or a bilayer. Archaea would also contain branched carbon chains attached to glycerol via ether linkages, and would now contain multiple origins of replication and different types of RNA polymerases. Furthermore, methionine acts as the initiator transfer RNA in contrast to 4-methionine found in bacteria. Now let's look at the archaeal membrane structure. Phospholipids found in Archaea contain branched tails attached to the glycerol component via ether linkages. In contrast, eukaryotic or bacterial phospholipids would contain unbranched hydrocarbon chains attached to the glycerol component via ester linkages. Some species of Archaea would now exhibit a lipid monolayer, whereas some would still exhibit the traditional lipid bilayer also found in bacteria and other eukaryotes. Recent studies, such as that conducted by Wang and others in 2012, suggests that the modifications on the surface of archaeal cell membrane prevents the entry of protons into the cell in highly acidic environments. However, up until this time, scientists are still trying to understand the archaeal cell membrane structure and how it functions even at extreme conditions. Species belonging to Archaea also contain unique cell wall structures. Remember, bacterial cell walls contain a peptidoglycan layer. This is also known as murine. This is composed of alternating NAG and NAM molecules. NAG stands for N-acetylglucosamine and NAM for N-acetylmuramic acid. Archaeal cell walls contain what we refer to as pseudomurine. Instead of NAG and NAM, it contains NAG and NAT. Similar to the peptidoglycan layer, the NAG and NAT molecules in Archaea are also linked by peptide cross bridges. Aside from the pseudomurine cell wall, some species of Archaea may also contain the S layer, which is a paracrystalline layer composed of interlinked proteins or glycoproteins. This helps stabilize your archaeal cell in extreme conditions. Moving on, members of your Archaea may be classified into different categories. Based on temperature, some may be identified as psychrophiles or thermophiles. Based on pH, some could now be identified as acidophiles. Based on salt concentrations where they live, they could now be identified as halophiles. Those that live under pressure are barophiles, while some would now be classified or identified as methanogens. Now let's look at some examples of members of Archaea that have been identified and photographed. First, we have Methanopyrus candleri, strain 116. It grows at a maximum temperature of 122 degrees Celsius, which is the highest ever recorded for members of Archaea. The species Methanococoides burtoni can live at 1 to 2 degrees Celsius and curiously up until 28 degrees Celsius. However, it requires methylated surfaces in order for it to survive. This shows that some members of Archaea can tolerate conditions which are not relatively extreme. For acidophiles, members of the genus Picrophilus can easily grow at a pH below 1. Another species of Archaea, Halorhabdus rutihensis, can grow at salt concentrations up to 27%. And indeed, there are other examples of the members of Archaea that have amazing capabilities and adaptations that allow them to survive in extreme conditions. Going back to the phylogenetic tree, you would now see one major group in your Archaea lineage is the Euryarchaeotes. Members of Euryarchaeotes, Poseidionalis and Pontarchaea, occasionally constitute a large fraction of the marine microbial communities. Members of the Sagmeg group or South African gold mine miscellaneous Euryarchaeal group are widespread in both terrestrial and subsea floor environments. Another major group in Archaea is the TAC superphylum. Members of TAC are readily cultivated and are therefore extensively studied. Cren Archaeota are among the first Archaeans that have been cultured and studied, whereas some Archaeota, which are known to be ammonia-oxidizing members of the TAC superphylum, are known to be the most numerically abundant Archaea on Earth. Another superphylum, the Asgard, was created after the discovery of Loki Archaeota. The first genomes of the members of this group were reconstructed from sediments taken near Loki's castle underwater vents in the North Atlantic Ocean. Soon after, other members of Asgard were discovered, and that includes Thor Archaeota, Heimdall Archaeota, and Hela Archaeota. To keep to the Norse god theme, this particular supergroup was named Asgard. Another large group under Archaea is the Deepan supergroup. This is short for the Apherites, Parvarchaeota, Enigma Archaeota, Nanohalo Archaeota, and Nano Archaeota. This extensively large group contains members which play critical roles in the cycling of nutrients on the planet. Now, why do we have to study Archaeans? Members of Archaea play a huge role in nutrient cycling and climate change. Some are able to produce methane, and studying such pathways would greatly help us address our methane problem in relation to the changing climate. Members of Microarchaeota and Parvarchaeota contain CRISPR-Cas9 viral defense systems, and this could now be further explored for biotechnology applications. Other biotechnology products may be derived from Archaea. This includes antifreeze proteins, enzymes which are stable at extremely high temperatures, and archaeal biological pathways may be studied, which could have important implications in medicine and research. And that is it for this video. I hope that you learned something about Archaea and that you'd be more interested in studying and learning more about it in the future. Thank you.